Hello friends, Ace Botany, next episode. In this episode, we'll be discussing molecular base of inheritance, second part. So we can directly enter to the topic. So I will share the screen, so it will be easy for you. I think screen came. So we can proceed. So chapter 5, molecular basis of inheritance, second one. And what all topics we'll be discussing in this episode? That is starting with introduction. Second is dogma of molecular biology. Third is packaging of DNA helix, both prokaryote and eukaryote. And as usual, 5 theory question, 5 NEET model question with the key. So we can proceed. See, as an introduction, so we'll be continuing with the DNA. And initially, certain thoughts in molecular biology according to NCERT. How DNA, such a lengthy molecule, arranged in a small place like nucleus in organisms. This is how the main topic goes today. And that is followed by question and NEET model question key and next topic. So how it will be organized? So we can start with the central dogma and reverse central dogma. Exam point of view, whether it is a theory or NEET type question, it is important. First, we should know what is meant by the term dogma. Dogma means an established suggestion or opinion. And when we come to central dogma in molecular biology, scientist is Francis Crick, 1958. I remember Francis Crick received the Nobel Prize in 1962 in connection with the structure of DNA along with Watson and Wilkins, etc. So what is central dogma? Here you can find DNA. Here replication DNA. From DNA, mRNA will be produced. That procedure is called transcription that we'll be learning later in this chapter. From mRNA, proteins will be produced. Again, that procedure is called translation, again coming in this chapter, last part. And which is the enzyme involved? Enzyme is important. Theory or fill in the blank and entrance. It is enzyme transcriptase. Interestingly, in central dogma, we can find unidirectional flow of information. I think that idea is clear. Diagram is simple, but theory and runs important. And in NCERT, there is a famous comment given here. In some viruses, the flow of information is in reverse direction. That is from RNA to DNA. Can you suggest a simple name to the process? This is a question from NCERT. Page number 83, March 24, reprinted edition. And its answer is this one. Here we'll be getting the answer. So two scientists, Temin and Baltimore, proposed a reverse of central dogma in certain viruses. And the viruses or the type where they worked was Rau sarcoma virus, RSV, single-stranded RNA. 1970, both of them done this work. And in 1975, they received Nobel Prize. Here, what is the procedure? We can find DNA, RNA, protein, that is same. But first, from RNA, DNA will be synthesized or that equation will be made. And that procedure is called reverse transcription. And from DNA, again, transcription, etc., that proceeds. Enzyme is reverse transcriptase. And this event is called reverse central dogma, otherwise refers to be Teminism. Name came due to the two famous scientists, Temin and Baltimore, in 1970. 75, they received Nobel Prize. I think both of this idea clear. It is important for theory and NEET exam, comparatively simple. So shall we proceed to next page? Now comes packaging of DNA helix. See, first we should know why there is a need that DNA has to be packed. Basic reason. See, when we compare the size of the nucleus and length of the DNA, then only we'll be getting an idea or an answer for it. Nucleus size is approximately 5 to 25 micrometer in diameter. Range will be there. Length of the DNA in a typical human diploid cell is that itself is a problem type question. NCERT given the two problem type question. So in typical human diploid cell, starting we studied genome, etc. You'll be remembering a table-like arrangement. NCERT 6.6 into 10 raised to 9 base pair. For converting into meter into 0.34 into 10 raised to minus 9 meters, an important calculation. And we'll be getting approximately 2.244 meters. See, one is micrometer, other is meter. Similarly, when we take prokaryotic cell like E. coli, length of the DNA, again, remember the number of bases, 4.6 into 10 raised to 6 into 0.34 into 10 raised to minus 9 meter. So it will be giving you a value 1.36 mm. So length is very much comparing with size of the nucleus. So length has to be adjusted in a nucleus. And nucleus size is just 10 raised to minus 6 meter average diameter. So you can guess how it has to be arranged. That is why such a heading came, packaging of DNA helix. I think that calculation is clear. It is important, direct NCERT theory and runs question. Now, DNA packaging in prokaryotes. We know that in prokaryotes there is no well-defined nucleus. But at the same time, DNA is not scattered. So practically DNA is seen in cytoplasm. So it has to be accommodated. So how things will be arranged? Nature made such an adjustment in which DNA is seen in supercoiled structure. And DNA is basically negatively charged, that is due to the phosphate groups in its nucleotides. The maintenance of these coils are done by non-histone basic chemicals like polyamines with a positive charge, so negative, positive. Total structure pack is called genophore or nucleoid. And DNA in nucleoid is organized in, next word is an important word, NCERT, large loops held by proteins. This is how nature arranges DNA in prokaryotes. In a supercoiled structure, then charge maintenance, negative, positive, and DNA nucleoid is organized in large loops held by proteins. I think prokaryotic arrangement is clear for DNA. Now, DNA packaging in eukaryote. Eukaryote, when we compare, it will be more complicated because cytoplasm, nucleus, etc. are separate. Wherever we go, that difference, that complication we can find. In eukaryote, nucleus, cytoplasm are separate. So automatically we can find complexities correspondingly increases, more area. The arrangement is carried out by a set of positively charged proteins called histones. Remember, DNA is negatively charged. So positively charged proteins called histones. A protein acquires charge depending upon the abundance of amino acid residue with the charged side chain. That is a line from NCERT page number 83. So an important question for theory and then like comment on it. That is why I'm telling NCERT is the base. But in discussion, we'll be slightly, little bit above NCERT because NEET exam point of view. Histones are water-soluble basic proteins. And histones are rich in basic amino acid, again important, arginine and lysine with the charged side chain. Here, 5 types of histones will be there, which include H1, H2A, H2B, H3, H4. And out of 5, 4 of them occurs in pairs together, forms histone octamer. You can find the diagram, histone octamer, otherwise called new body. The positively charged histones octamer is surrounded by DNA. See, this is DNA, this is histone octamer. So diagram-based question, previous NCERT diagram was different. Here, new NCERT diagram, previous one different. And here you can find linker DNA, that is H1. I marked here, and this red is nucleosome, total assembly. Histone octamer is alone called new body. Total structure is called, that is positively charged histone octamer surrounded by DNA forming nucleosome. This is another NCERT diagram. And here you can find certain beads. That is a usual question that bead stands for. Bead stands for nucleosome. Nucleosome means entire structure, positively charged histone octamer surrounded by DNA. I think that is clear. Again, important for exam point of view, theory entrance, NCERT topic. Now we can proceed to next area. How this will be arranged. It's very interesting. So old NCERT diagram, this one. Here we can find DNA with a 2 nanometer. You'll be remembering 20, 10, all this value will be learned in first episode. The nucleosome, 10 nanometer, 5 times more. The histone octamer you can find, linker histone you can find. This is solenoid. Solenoid is 30 nanometer. Physics will be learning solenoid what it is, just like a springlex. Then here it is super solenoid. Super solenoid is 300 nanometer and it is chromatid with the 700 nanometer, chromosome with the 1400 nanometer in size. This is how that organization takes place, how much compress, how much coiling we can find here. See, the nature can do anything if it likes. Now, various steps in the foldings and superfoldings of the basic chromatin components to generate an eukaryotic chromosome. This is the heading for these diagrams. Present NCERT, I think this diagram is not there. It's very clear, easy to follow. Size comparison we can easily make. Again, match the following type question. Each item DNA, nucleosome, solenoid, super solenoid, chromatid, chromosome, and the bottom values will be changed and give correct to match the following correct one. Fill in the blank type question. I think idea clear. Now, a small problem connected with NCERT. How we can make an approximate calculation. In a bead, connected with the nucleosome, there will be 200 bases. 146 plus 54, 200 base pairs. So when we come to mammalian cell, this value will be familiar, 6.6 into 10 raised to 9 base pairs into 1 by 200, because 1 by 200 means in a nucleosome, 200 base pairs. So that will give you 3.3 into 10 raised to 7 beads. So in mammalian cell, approximately 3.3 into 10 raised to 7 beads will be there. This is NCERT question, page number 83. Same with the Escherichia coli, you can calculate. Both of them, theory question, entrance question, not together. Either one or other will be usual question. I think idea clear. And when we come to higher level packaging of chromatin, it requires additional set of proteins called non-histone chromatin, NSC proteins. Different types are seen. Now we are proceeding to, I think this idea is clear, important, simple, don't leave it. Now when we proceed to next page, here we can find. So 2 types of chromatins we can find. Another usual question. 2 types, very predominant question, exam point of view, theory entrance. And how these types of chromatin has been separated or identified. It is based on staining nature in a typical nucleus. The comparison between the 2 types of chromatin for us, that is euchromatin, heterochromatin, either table-wise question, comparison question, find the odd one, all type of question can come from this area. So this episode is important for exam. So first type is called euchromatin and second type is called heterochromatin. What are the difference between these two? Clearly I written this, easy, just like hardwood and sapwood. This chromatin is loosely packed. Which one? Euchromatin. So correspondingly there, the other side, densely packed. Euchromatin is transcriptionally active. So here it is inactive. Then in euchromatin, its replication takes place in early synthetic phase. So less time for unwinding. Here it is delayed because replication takes place in late synthetic phase. So it takes longer time to unwind. Now euchromatin stains lightly. Heterochromatin stains darkly. So euchromatin staining will be lightly or slightly. Anyway, we can use heterochromatin. It is darkly stained. And euchromatin is genetically active. Heterochromatin is genetically inactive. So this difference is an important question for theory entrance NCERT base. And our next topic basically includes or starts with the search for genetic material. More and more experiments, again, exam point of view, all these areas are important. And after that, 5 theory question and 5 NEET model question with the key exit. So what all things we learned in this episode? We can just pass through it. First, we introduce the topic, then introduction given, connected with the DNS extension, then central dogma, reverse central dogma, central dogma proposed by, what is the idea? Then after that, NCERT asking what is happening in some viruses like Rao sarcoma virus, where Temin and Baltimore proposed, reverse central dogma. After that, we entered into packaging of DNA helix. Reason is length is too much in comparing with the size of the nucleus. Then how it has been packed. Two problems given here. 6.6 into 10 raised to 9 base pair into 0.34 into 10 raised to minus 9 meter. That is 2.24 meters. Other one equally 4.6 into 10 raised to 6 into 0.34 into 10 raised to minus 9. So that will give you 1.36 millimeter. So length is too much, where size of the nucleus is comparing with the length, it is very small. So it has to be arranged. Prokaryotes, since no nucleus, how it is accommodated, that is by supercoil structure. DNA is negatively charged due to the phosphate groups in nucleotides. And the maintenance of these coils is done by non-histone basic chemicals, polyamines with a positive charge, so DNA negative, this one positive. Total structure is called genophore or nucleoid. DNA nucleoid organized in large loops held by protein, large loops. Now DNA packaging in eukaryote, more complex. And arrangement is carried out by a set of positively charged proteins called histones. And how a protein acquires charge depending upon the abundance of amino acid residue with a charged side chain, NCERT line. Histones, what are they? They are water-soluble basic protein. Histones are rich in amino acid like arginine and lysine, which is basic with a charged side chain. 5 types of histones: H1, H2A, H2B, H3, H4. H1 is called linker histone, and other one core histone. 4 of them occurs in pairs. Histone octamer or new body will be formed. Then positively charged histone octamer is surrounded by DNA. It is forming nucleosome, and this diagram is important. Beads, then DNA, linker histone, and here you can find linker DNA as well as nucleosome. I think idea clear. Then we entered into how it has been arranged. Starting with the DNA, nucleosome, solenoid, super solenoid, chromatid, chromosome, 2 nanometer, 10 nanometer, 30 nanometer, 300 nanometer, 700 nanometer, 1400 nanometer, and how this has been arranged. Then calculation again. 1 bit, 200 base pairs. So 6.6 into 10 raised to 9 base pair into 1 by 200. 1 by 200 means 1 nucleosome, 200 base pairs. So 3.3 into 10 raised to 7 beads. So in mammalians, there are approximately 3.3 into 10 raised to 7 beads, NCERT question. And packaging of chromatin high level requires additional set of proteins called non-histone chromatin NC proteins. Different types will be there. Then comes another important area, different types of chromatin based on staining nature. Euchromatin, heterochromatin, then difference between these two. After that, what will be the next topic starting? Search for genetic material, then 5 theory question, 5 NEET model question with the key. Hoping that it is clear. If you like this episode, subscribe or like it, share it, comment on it. We can meet in the next episode. Take care. Thank you.